Outstanding.
Perfect. So you may ask why I'm just next to Hans like this. We're not on the Fireside Chat, and why Dr. Topalian and Pardoll are separated. Well, I think COVID had different, plans for Dr. Pardoll, but, COVID cannot stop these two cancer trailblazer and immunotherapy trailblazers here. So we're going to continue, despite the email we got was at 3:21 a.m., the Fireside Chat here. And welcome you, Drew and Suzanne, good friends of the kidney cancer community and the immunotherapy and the cancer community in general, to KCRS22. And I'm going to let your former colleague, Hans Hammer, introducing you.
Well, thank you so much, Tony. So again, I have very fond memories of my time at Hopkins, and when it came to think about another, power couple, tycoon couple in immunotherapy, it was very clear that we should approach Drew Pardoll and Suzanne Topalian, who are really leading the immunotherapy research efforts at Hopkins. Drew, as he will probably point out, he's a Hopkins product through and through, if I remember correctly. And he founded the Bloomberg Kimmel Immunotherapy Institute. He is the Abloff Professor for Oncology across different departments. And he directs really the cancer immunology program at the Sidney Kimmel Cancer Center. And he's known for discovering numerous targets, PDL2, LAG3, has done a lot of work in T regulatory cells, and his work has really infiltrated, so to say, across the different disciplines at Hopkins to really move the field forward. When I was starting out my work at Hopkins, we went from very little work to the time when these drugs were first in clinical trials, and Suzanne Topalian picked it up and trailblazed forward with this. She is the Bloomberg Kimmel Professor of Cancer Immunotherapy at Hopkins. She directs the melanoma program there and has been awarded with numerous awards, including the Konofsky Award, and also was elected to the National Academy of Medicine. But enough of that. I could spend a whole 45 minutes praising them for their work. We're here to learn about them and learn a little bit about the story, and really, with that, inspire the next generation of people who will help us hopefully move the work forward in kidney cancer. So I thought, let us get started with learning a little bit more about yourselves. Why don't you tell us a little bit about maybe like where you grew up and how you eventually ended up in medicine in general.
Maybe we'll start by Drew.
Yeah.
Okay. I grew up in Elizabeth, New Jersey. Was actually born in Newark, but grew up in Elizabeth, and became very interested in two areas. One was astronomy, because I got a telescope for an early birthday gift when I was young, and the other was biology, because I got a microscope. And ultimately, I was actually a chemistry physics major at Johns Hopkins. Hans is correct. I've been at Hopkins essentially my whole career, and I've had one job in my life. They changed my position title occasionally, but and I ended up in part because one of my mentors, teachers in college, developed kidney disease, in fact, not cancer, but secondary to juvenile diabetes at a time when there was far less that one can do. And he eventually succumbed to his renal failure. And so that ultimately moved me in the direction of medicine. And I actually became interested in cancer because I ran into an amazing person that Hans knew, Don Coffey, who is a urologist. And I decided to do cancer immunotherapy when it wasn't fashionable. when I was actually a senior resident on the bone marrow transplant program. This was pre-cyclosporin A days, where graft versus host disease for allogeneic transplants was really, really nasty. But you could see in front of your eyes the power of the immune system. And we used to tell the patients that if you survive your graft versus host disease, your cancer won't come back, because there is this graft anti-tumor effect, or graft versus tumor, that was known from mouse studies from Korngold and Sprint, was mediated by T cells. So I decided that if we could learn to control and target the immune system more effectively, this should be a great anti-cancer weapon. And that's what I've been trying to do for the last 35 years.
Beautiful. Yeah, what about you, Suzanne?
Yeah, so I also grew up in New Jersey, and in fact, just about 15 miles away from Drew, but we didn't know each other in New Jersey. We actually met at a scientific conference in Taos, New Mexico, but that's another story. So I had a hard time deciding between whether I wanted to go into the arts or science when I was in college. So I majored in English literature, but I did all my pre-med courses at the same time. But when I started college, actually in the early 1970s, was when Nixon, who was president at the time, declared war on cancer. So everybody was interested in that. And this appeared to me to be the new frontier, curing cancer. So I was on board with that. I decided to go to medical school. And I decided to train in surgical oncology. So I did my surgical residency at Jefferson in Philadelphia, where you are now. And during that time, I spent a year of research at the Children's Hospital of Philadelphia, and that's where I really caught the bug with my interest in immunotherapy, because I was in a cancer immunology lab just because I was interested in working with the surgeon who ran the lab, but I really got hooked on the idea that the immune system should be able to reject cancer the same way that it rejects viruses and allografts. So after that, I decided to go down to Bethesda and do a fellowship with Steve Rosenberg, who even at that time was a preeminent cancer immunologist and was running immunotherapy trials. And stayed down there for 21 years. Actually, I was a senior investigator there, and then moved to Hopkins in 2006 to work on anti-PD-1. And you may remember that 2006 was the year when the very first patient was treated with any anti-PD-1 drug, which happened to be nivolumab. In an all-comers trial where renal cancer was included. So that's my story.
I mean, that's perfect. Now, I have to tell you, with all these fireside chats, I mean, Hans has been asking the professional medical question, and I have been asking the non-medical question, but you alluded to, so, we did not spare another power couple a few years ago, Gordon Freeman and Arlene Sharp. But how did you meet, and perhaps any memories from the first date? But we'll start by Drew.
Yeah, start with Drew.
So actually, it's great to follow after Gordon and Arlene, who are actually good personal friends. Not too long after we actually met it was actually literally over a slide projector back then, because we were speaking in the same session, which in Taos, New Mexico. I wanted to ask Suzanne out on a date, but I was a little bit timid. So then I was actually asked to chair and be able to invite people to a small conference at Hilton Head, which I accepted. And Suzanne was the first person I called, half because I knew that she would be a great person for the conference, which was on immunotherapy, but the other half was it was an opportunity to ask her out on her first date, which actually she was on crutches because it turned out that a day or two after I'd left the Taos meeting, she had a skiing accident, but she said, happy to go out if you don't mind my being on crutches. But on our first date, she lived in a tall apartment very near the NIH. And I got the floor wrong for her apartment. So I knocked on the door, and this very lovely elderly couple answered. And so for the next hour, they were trying to figure out, because in the evening, there wasn't anybody at the desk, what Suzanne's address was. And so finally they found it. But Suzanne thought that I had stood her up. But after that mishap, it was all uphill.
Is that your take, the same take, Suzanne, or
Yeah.
Drew tried hard?
Yeah, no, that's my take. I thought I had been stood up, but he got there just in time.
Okay, and I guess he let you walk on crutches, didn't hold you down the stairs or anything.
Right. Yeah.
Okay,
Yeah. Things were good from that point on. Yep.
that was great. That's a great story.
That was actually 30 years ago.
Wow.
So we would love to invite the audience to maybe ask a couple of questions. We'll look at the chat, but let us know if there's something. In the meantime, I will collect some of those. I remember, for example, when I went to one of your holiday parties, or in general terms, I think for both of you, I think mentorship is something that you take very seriously. Maybe you want to talk about how essentially the way I remember it at Hopkins, you were able to really collaborate with so many people, draw so many people into immunotherapy that the program became what it is. And like, how do you go about this, and maybe some of the way you think about mentorship?
So mentorship is a very important issue. And, now at Hopkins, every young faculty and even our fellows have mentorship committees to be sure that they're able to maintain focus, that they have clear goals in mind for their careers, and that they're getting the appropriate support that they need at the university. But 20, 30 years ago, these things were not in place. And so people had to find their own mentors. You might imagine training in surgery as a female in the early 1980s. There weren't a lot of female mentors who were, professors of surgery at that time. But I had wonderful mentors, and I think my attitude is that you don't necessarily need to have a mentor who is exactly like you, in appearance or whatever. You need to find people who are on the same wavelength in terms of what you're thinking about doing with your career, and people who are going to be very supportive. And so that's the advice that I give to young people, is don't be too fixated on finding somebody who looks like you. Just try to find somebody who thinks along the same lines and who is going to be there for you when you need support.
Drew, anything to add to that? And one thing I'm interested in, have you ever mentored some people in common, or you keep things different, or people jump from your lab to Suzanne's lab back and forth? How do you approach mentoring?
Yeah, well, first of all, I'll say that mentorship is, I think, in a lot of ways in the DNA of Johns Hopkins. I can only speak for Hopkins, but that's very important. And I've certainly had many great students and fellows. Just to add to Suzanne's comment about, you don't have to find a mentor that necessarily looks like you or whatever. I actually had many important mentors. For my PhD, I was actually one of the first graduate students to have 2 co-mentors. Back then, that wasn't a thing. Now it is. Don Coffey and Bert Vogelstein. Actually, Bert was a junior faculty member. I was his first graduate student. Don was senior, and you could not find 2 people with more different styles. And so it was a valuable process for me to sort of ask, what do these guys have in common? I mean, they were both incredibly successful, and one was obviously a passion to understand cancer at a level that actually allowed us to develop new treatments for cancer. And the second one was the appreciation that the engine in the laboratory were the students. And so I think, I think these are very important. Suzanne and I have both separate projects, but also interact on a lot. You can see this by our publication list. And certainly, while there aren't many folks that we necessarily formally co-mentor, there are a lot of people that are back and forth between our labs, and a lot of a tremendous amount of interaction. So there's a certain seamlessness to the environment of discovery and mentorship, I think.
Wonderful. We have a question actually coming in from a long-term survivor from IL-2, and this probably goes in the direction of Suzanne's experience. And the survivor asks, sounds like, she's maybe a little bit disappointed that high-dose IL-2 is not as frequently anymore. And the question goes a little bit in the direction of, how can we utilize IL-2, maybe a similar compound, so to say, in this era of immunotherapy?
Yeah, so being at the National Cancer Institute, I was a co-investigator on the earliest trials of Interleukin-2 in kidney cancer. And I'm very glad to hear about this story. I mean, we know that maybe 12 to 15% of patients with advanced kidney cancer have a major response to Interleukin-2, and that those responses can last for a very long time. And I think we have to assume that after so many years that there are true cures for a percentage of patients. But what hampers Interleukin-2 is that really it was only effective at high dose. We tried to dial down the dose to make it more tolerable, and the response rates were not as good with the outpatient regimens. So this is a very intense regimen with a lot of acute side effects. We have things now that are safer and still can offer advantages and even to higher percentages of patients, such as anti-PD-1 treatment and the anti-PD-1 combinations, which are now used very commonly in kidney cancer. But we still reserve high-dose IL-2 for patients whose tumors do not respond to these newer immunotherapies. It is still available, but now it's more like maybe a third line or later treatment option. And I would defer to Hans and Tony to say more about current practice.
Yeah, the one thing I want to add, and maybe you and Drew can comment, is that all the attempt to use other form of IL-2 after, high dose, intravenous, subcutaneous, or others, including more recently the data with pegylated IL-2 that do have an affinity for some receptors, the alpha, I think, that exclude the alpha, have not panned out in randomized studies. And we still have a lot of investments and companies going the route of cytokines, which is totally fine, more options for our patient. But maybe Drew, do you think, what is the next big thing in cytokine, and is it still the focus on IL-2?
Yeah, so I think with regard to IL-2, I was never a big fan of the pegylated IL-2 because I think I was never convinced about that it selectively concentrated in tumors, and also that it was selective for the CD8 killer cells, which have predominantly the beta gamma IL-2 receptor versus, as you said, Tony, the Treg cells, which you don't want to activate and expand, which have the alpha beta gamma. However, there are newer, really much more sophisticatedly engineered mutants of IL-2. Actually, a fantastic young researcher, Jamie Spangler, has developed a number of these that, based on crystal structure, really hit either the alpha beta gamma, and those she's actually interested in to expand Treg cells to treat autoimmune disease, versus the ones that hit beta gamma, but not alpha beta gamma, which don't hit Tregs at all and can activate. So I am not ready to give up on some of these, I think, much more properly engineered forms of IL-2. With regard to other cytokines, I think the challenge has always been that if you look at the biology of cytokines, they're really designed to work within about 5 or 6 cell diameters as a communications mechanism of the cell that's producing them. Then their concentration falls below that, which will trigger the receptor. So geographical localization is sort of built into their natural biology. When you flood the whole body by giving huge IV doses, then you just really mess up that physiology. The one potential exception that we're actually very enthusiastic about is IL-7, which is an interesting cytokine that maintains not only naive cells, but memory cells of the sort that may really sustain anti-tumor immunity. And it turns out that it's actually not that toxic at all given systemically. So if you had to ask me for one that I have enthusiasm for systemic, I would say IL-7. And then the other part is using antibodies or other means to try to target cytokines really more selectively to tumors, which again, the PEG IL-2, I don't think really did that. So anyway, just
we do have,
I think there's more to come with these.
We do have a lot of patient advocate and scientists, immunotherapists asking questions, but we're gonna, since we still have 15, 20 minutes, one of the question came from Dr. Nirmala. You wanna say it, Hans?
Yeah, so what about if you both are interested in a certain topic? Do you compete on grant applications? How do you handle potential conflict of interest there?
No, that's where the multi-PI R01 comes in handy.
Yeah, when we actually have a big multi-PI grant together, Suzanne, myself, and Janice Taub, who did a lot of the early work on the immune microenvironment of tumors. So I wouldn't compete with an English major because she writes far better than I do.
100%.
What about some of the other targets that people are thinking about? I know you've done some work with LAG-3, and LAG-3 seems to have some nice effects in melanoma. So immune checkpoints, also CAR T cells. Like, what is your thinking there? where things might be going on your impressions? It sounds like some of these topics are difficult, like CAR T cells and solid tumors are somewhat challenging, it seems, right?
Right. I mean, the LAG-3 actually is in testing for kidney cancer, and of course made a big splash in March with the FDA approval of a combination of LAG-3 and anti-PD-1 for metastatic melanoma. So in that case, in a randomized phase 3 trial, there was a significant improvement in progression-free survival in patients with advanced melanoma. So LAG-3 combinations are being tested in other diseases like kidney cancer. They're also being tested in patients with earlier stages of disease, such as in the pre-surgical or neoadjuvant setting, or in the post-surgical adjuvant setting for resectable cancers. So more to come on that. And certainly there's so many other checkpoints that are in clinical testing, but right now the three that have across the threshold to FDA approval would be PD-1, CTLA-4, and LAG-3. In terms of CAR T cells, so the other part of this question, of course, in general, it has been very difficult to use CAR T cells safely in solid tumors. So as we know, they're very effective against blood cancers. But the issue with solid tumors is finding the right target for the CAR T cells that will not, lead to a lot of side effects. So far, we're not there. Kidney cancer, though, does have some unique opportunities for targets, such as endogenous retroviruses. Maybe I'll let Drew say a bit about that because he's worked on this.
Mhm.
Yeah, so just with regard to the endogenous retrovirus, and there's interest in those actually in kidney cancer, which is one of the interesting exceptions if you look at sort of that straight line curve comparing total mutational burden to response to anti-PD-1. Most tumors fall on average on that curve. The three exceptions really are Merkel cell, which is really because of the Merkel cell polyomavirus, mesothelioma, and kidney cancer, which has a relatively low burden of classic, single base mutations, and we still don't understand why its response is actually, falls above that curve. I think it's actually very important to understand that. These certain endogenous retroviruses, which are sort of expressed de novo in cancers, I think are a great target. I think at the broader level, while I think T cell, there's a lot more T cell checkpoints as targets. I think that myeloid cells, in particular macrophages, myeloid-derived suppressor cells, which are distinct but linked myeloid lineages, they're, I think, going to offer a great opportunity. Many labs, including ourselves, are looking at macrophage-specific targets, and I think there's still modest activity. There was disappointment with the CSF1R inhibitors, but that's just one. I think that's going to be a big area of interest.
One question that came through the chat, which is very, very important and very relevant to KidneyCAN. KidneyCAN is a grassroots movement where investigators, patient advocates, initially the Department of Defense, all set together an agenda that we are going to cure kidney cancer in this lifetime, and we have to do it with partnership. And of course, one of our most important, biggest partner are the industry and the pharma company. Question from Dr. Suzanne. who recently joined us at Dana-Farber. How do we keep this relationship between academia and pharma productive and focused on patient-centered outcome?
That's a tough question, but I think it all depends on dialogue. And so, as much as possible, I think academics need to reach out to pharma and vice versa to create a productive dialogue around these issues. In academia, we know that there's only so much we can do in terms of early phase clinical trials after doing a trial that might have 20 to 50 patients. When you start thinking about taking something to phase 3, you have to engage pharma. I think there's no way around that. And a lot of it depends on the personalities involved. I mean, I think I've been very lucky along the way to be able to interact with people who were focused on patient outcomes. So that worked. But it's a challenge, but it is a goal that we all have to keep in mind.
Drew, anything to add?
Yeah. We could spend a whole day on this. And I like to say about pharma, can't live with them, can't live without them. The one thing I would add is biotech to this. I think there are really three. The biotech is the third leg to the stool. Biotech is all about taking somewhat bigger risks and on average is closer to academia. There are now more and more examples of academics jumping to pharma, but I would say that up until 10 years ago, the pathway was mostly academia to biotech. And then biotech takes something a certain distance and then pharma picks it up. That's what happened with PD-1. I mean, we went to Medarex. Nivolumab was originally MDX 1106. And we actually started collaborating with them in 2000. Then one day, Medarex was BMS in 2009. And it took, I think, maybe a year and a half to 2 years for us to learn how to collaborate with pharma and vice versa. But that was a great collaboration. And again, this comes down to relationship building, because ultimately, even in pharma, when you look at programs, LAG3 was an example of this, you need champions. Quite frankly, BMS was going to ditch LAG3 because it didn't have single agent activity. But we had shown that in combination with anti-PD-1, it did better than PD-1 alone. And so there really was synergy. There were some very key collaborators who became friends, people like Nils Lundberg, Fuad Namouni in BMS, that we got together with at meetings and built the relationships. And they were the advocates as part of that relationship within the company. So I think, yeah, this is critical. And then the final thing is that the power of advocacy is absolutely incalculable. And the reason for that is that when we advocate for more funding, whatever, we have an inherent conflict of interest. I mean, we need the money to do our research, but we also need the money to keep our jobs, because as Hans knows, when you work at an academic institution, it's a hunting license to write grants and whatnot. Whereas advocates, really don't have that kind of conflict of interest, and have unbelievable power and influence, including in biotech and pharma. And I would actually advocate that advocates focus on that and focus on things that pharma can do to be more innovative.
Well, we brought in, Susan Petit, one of our star from day one, kidney cancer advocate, who's here, who had a big part in KCRS22. And, I want to re-emphasize to everyone joining us by Zoom or in the room that look at the Department of Defense, the CDRMP. Kidney cancer was not on the map. We started with lobbying with kidney cancer advocates with 10 million, and now we're up to 50 million. The young generation, or all the old geezer like myself, who benefited from that and lured, in a way, not to use other word, folks to kidney cancer is amazing.
Yeah, I totally agree. It's really all these different legs of many different legs of a stool that need to come together to make a difference, from advocacy, pharma, academia, biotech. So somebody starting out today, the hunting license to go after grants, sometimes in a fragile environment where the first 1 or 2 grants are critical to survive as a young academician. What kind of advice would you give these hungry but young and maybe scared people how to go about it and start it?
Well, I would say be persistent. Even us old folks know that you may not get your grant funded the first time. And so you just go back, listen to the reviewers, take the good and ignore the irrelevant comments and improve it and go back. I think coming full circle to our discussion of mentorship, this is really where mentors are very vital. Because I know there are a lot of young investigators who spend a bit too much time sort of carpet bombing every opportunity, no matter how slim, to get funding. And I think there needs to be some focus to it and prioritization on things that are most likely to be productive. And that is really, I think, where the mentors come in.
How about you, Drew?
Yeah, so I have actually not a lot to add. I agree completely with Suzanne. And one of the things I say is that the most overrated thing when you take your first job is the package, and the most underrated thing is the environment. And the environment really is about the mentors, but also having diversity of expertise in an environment in which, senior people with various expertise are motivated to help junior people.
I think that's perfect. We can go for hours and hours, but, I cannot thank you enough for this. I want the audience to know we need to take, 5 minutes. You made it happen after, the reason, and we said there is no way we're gonna cancel this Fireside Chat. So maybe last few words of wisdom, starting by you, Suzanne.
I would just say how much we're impressed with KidneyCAN, with the energy and the passion, the involvement of people from all sectors, including the advocates. It really does take a village. So I guess that's my word of wisdom is it takes people from all sectors working together to move this forward. And I think you all are really doing that. And it's been so great to meet you.
And Drew?
Yeah, I would just steal the Wayne Gretzky quote when he was asked how he was the greatest, despite the fact that he wasn't the fastest skater, the strongest, or the biggest. And he said, that's because I don't skate to the puck. I skate to where the puck is going.
Oh,
Very good.
this
very
is
good.
very, very good. Well, thank you. Thank you so much. We're gonna now go downstairs. Drew, please feel better.
Yeah.
And hopefully we'll stay in touch and continue to make, you know, new headways in kidney cancer and immunotherapy.
Thank you so much, Drew. Thank you so much, Suzanne.
Thank
Thanks so
you.
much.
Bye.
Take care, everybody. Enjoy the rest of the meeting.
Thank you.
Great. Thanks, everybody. And we're not going to do a break right now. We're going to call up our next two moderators and their panel for session 1 on metabolism. And they're going to be led by co-moderators, Dr. Mehdi Malapur from State University of Upstate New York and from Maria Carlo from Memorial Sloan Kettering. So thank you all for moderating.
Thank you. Right. So can we have, is this on? Thank you. Can we have the speakers in here so we can test the mics? You want to sit here?
Sure.
Sure. Is there anything that they need to, you're good? Mic check for everybody. Mic check.
Yeah, can I just get everybody to introduce themselves by way of a mic check, and I can get the online people just to check that they can hear everybody well.
Okay, can I say a few words before we get into the introduction?
Yeah,
Okay, wonderful. So my name is Mehdi Malapur. I'm professor of urology at Upstate Medical University. I have my dear friend here, Marie Carlo from Memorial Sloan Kettering to chair this session. But before I start the whole session, which is about metabolism and mechanisms with focus on health disparities, we have, is that the camera that is connected to the virtual? So I want to say welcome to everybody who's joining us virtually. I know last year I was there and I felt like I was being ignored. So we see you, we have eyes on you, we are looking at you, so we see what you're up to. So welcome. This meeting was an amazing session this morning, and what a heartwarming story. You know, I hope one day my wife, you know, Dr. Borboli and I, we can come here and share. Yeah, we can throw big names, you know, Dr. Marsden and I, we can throw in big names. So before I proceed, first of all, can we give a round of applause to Mr. Brian Lewis and Susan Pichi? I know we have superheroes, but these two are my superheroes. So we are going to have very small talks, and the thinking behind this session was we went through all the abstracts that were funded, all the grants that were funded by the DoD, kidney cancer, in 2020, and we selected these abstracts, again, my dear friend, and we narrowed it down to a few talks that we thought is very fitting for this session. Unfortunately, Dr. Adam Matwali cannot be with us today. He has a personal problem that he could not join us, and instead, Dr. Mas Owl, if I pronounce your name correctly, he's joining us to do this session. Without further ado, let's start, and I will hand the mic to my friend. We can start.
Good morning, and thank you for the introduction. I was going to introduce our speakers, but I guess they're introducing themselves as way of mic check. So, Ken, we can start with you.
I'm Ken Batai. I'm from Roswell Park Comprehensive Cancer Center.
I'm from UNC, and I'm the postdoc from Dr. Qin Zhang, and I research virtual signal pathway.
Hi, I'm Faize. I work in Dr. Brugarlis lab at UT Southwestern, and I'm a postdoc.
Hi, I'm Pavlos Misawel from MD Anderson.
Great, thank you. So as Mehdi was introducing, we'll be talking, this session includes DoD-funded projects that talk about mechanism and metabolism with a focus on health disparities. In particular, we'll be focusing on non-HIF mediated novel targets. And then metabolism of obesity, and then metabolism of hypoxia and renal medullary cancer. So I think we'll start with Ken's talk. So the format for this will be 7-minute short talk and then 7-minute discussion. So I think we have mics on tables. Oh, over there. So our virtual audience can hear us. And then we'll be also seeing simultaneously questions through the chat. So hopefully we'll have plenty of discussion. So 7-minute talk, 7-minute discussion.
All right. So I'm going to see my slide here. Or
Yes,
I'm not sure.
I hope so.
All right. I'm waiting. Oh, it's over there.
So the slides are not showing up here, so I don't know if you want to.
Okay, I'll stand up so I can see my slide a little bit. So I'm from Department of Cancer Prevention. So I do a lot of prevention research. So it's a little bit different from a lot of you guys. And I'm a disparity researcher. So I'll be talking about disparities in Hispanic population, particularly. Is this the one to switch?
I think so, sorry.
All right, I don't know how to work this. Okay, here's one.
What do you say, next slide?
Yes.
Okay.
Next slide.
All right, so just quick background. I've been using a lot of different data sources to documenting disparity in Hispanic American, American Indians. So I've done a lot of research in Arizona where I was till recently. So quickly to summarize, Hispanics and American Indians are more likely to have a higher kidney cancer incidence and mortality, earlier age of diagnosis. They have predominantly clear cell subtype. They are often diagnosed with advanced stage, and they tend to have more invasive treatment, so tend to be radical rather than partial nephrectomy for the stage 1 cancer patients. And as I was going through my data, I noticed was that rate of obesity and diabetes really high in the Hispanics and American Indians, our patients. So also the clear cell type is obesity driven related clear subtypes. So I was looking into more of the research. Next, please. This one? Yes, okay. So last several years or so, people talking about structural inequality and all those. So I look at quickly about how that structural inequality is related to kidney cancer patients. As you can see, as a patient has more coming from the more disadvantaged neighborhood, they tend to have the obesity rates tend to be higher, which is really significant for Hispanic population and Native Americans. But for the Hispanic population, actually, you adjust for the social deprivation index, which is linked to neighborhood factor, and that obesity is accounted for that. But American Indian is still significant. So this is what I started thinking about as I developed into my research. So there are 3 different patients, and the first one is young Hispanic patients, male, obese, and he might be really different from all the white patients who are skinny or young female patients who are also skinny. So that's a question we have, and do they have a different type of kidney cancer? So first step, we wanted to look at the body composition difference across between Hispanic and non-Hispanic white, and stratified by sex. Here's showing visceral fat is actually lower for the non-Hispanic white female. We see a difference in the male and female. That's pretty much well documented out there. But for the non-Hispanic white, visceral fat is very low, which is very different from Hispanic female who has a high subcutaneous fat. And we saw that correlation for the subcutaneous fat and the age, so the young patients tend to have a high subcutaneous fat. And when we look at hypertension or diabetes, So patients with hypertension, diabetes tend to have high visceral fat. And then we are looking at gene expressions. So CCB tend to be, patients with CCB has a shorter time of survival, which is well documented in other studies. So we are thinking a Hispanic patient may have a CCB, but it was actually opposite in a way that CCA, I think this obesity adiposity related subtype is more common in this Hispanic patient. And we are going through the next step, stage of analysis using a urine and the plasma sample and trying to show that if there's a difference between the Hispanic patient and non-Hispanic patient. At this point, we don't know if they are obesity related or diet or anything about this or how they are related to stage or grade. So that's something we will be doing as we go from the next couple of years. As I say, most of the research was done in Arizona and supported by DOD and also by AUA and NCI, ACS, a lot of grants, fortunately, yes, thank you.
Great. Thank you. So we will open to audience questions, and then we'll stay tuned here for questions from the virtual audience. So I can start with a question. It's interesting because when you look at epidemiology of cancers, and recently the statistics for cancers in Hispanic Americans came out, and there's a clear difference, for example, lower rates of lung cancer than you would expect, but higher rates of other cancers like endometrial and colorectal, which also may be related to obesity. Is there any research going, I don't know if you've looked at or others of that type of research in other cancer types and how that may apply in kidney cancer, the role of adiposity in cancer in general?
What I think is, first of all, when we're looking at Hispanic, we try to separate between Mexican Americans, that's what we have in Arizona, might be different from Puerto Ricans and Cubans, so just want to make sure that is a first step we think about. And obesity rate could be different between Hispanic subtype. And in Arizona, we see a lot of obesity-related cancer is higher in Hispanic community, also American Indian communities. So, for example, we are looking at the liver cancer, which has another obesity-driven cancer. It's very high in Hispanics and American Indian compared to non-Hispanic white, and they have high mortality rate as well.
So there's a question there if you want to.
How much of disparity might be related to intratumoral metabolomic difference and compared, I couldn't.
We couldn't see the rest of the question. Right. If you can put the Q&A. Go ahead, Tony.
So here the ancestry, this was self-referred and self-reported race, right?
Yes.
Have you thought about, if you have samples, to look at genetic ancestry and see if it correlates with it or how that is a correlation, but it's not perfect, obviously.
Yeah, definitely. So that was
if we can maybe repeat the question for the virtual, I'll just say Dr. Chueri had a question about genetic versus self-reported ancestry and how that may differ in terms of outcomes.
So definitely, so like Hispanic American in Arizona, mostly predominantly Mexican American, generally has a high Native American ancestry. So we are really thinking there's some similarity in the genetic background. And we have done a little bit of study looking at genetic ancestry and seeing Native American ancestry seems to be correlated with the younger age of diagnosis, but that's the next step I'd like to take. We're going to collect more samples. Well, I have a sample, so I just get a little bit more money to genotype and sequence and get answers to the estimated and figure out the correlation with clinical characteristics.
So we had the last question, but it disappeared. Okay, how much of this disparity might be related to the intratumoral metabolic differences compared to extratumoral milieu, for example, visceral fat?
Yeah, another question that I really wanted to explore from R01. So it's just, I have no idea at this point, and that's something I wanted to look at. We're collecting a fat tissue surrounding that kidney to just maybe do the gene expression or metabolomic analysis to see if there is a difference between any metabolic signature for Hispanics and white patients and accounting for so many factors.
And one last question from the chats. Any information about the microbiome differences in these populations?
I would assume so. I haven't looked at it because, diet different, obesity different, and I assume there should be some kind of microbiome difference. And we could look at the urine microbiome if we want to. Just, yes, again, I think it's something to do in the future.
Okay.
Great.
Let's give him a round of applause, please. Thank you. I feel like I'm your co-pilot, but I'm taking over your session. I'm going to be quiet. I have, it's a bad habit of mine. Please.
So our next speaker is Shizhuan Liu. No? I think so, no? We'll see. Oh, no, Faize, Sachi, sorry. You're right.
All right. So my name is Faize. Like I said, I'm a postdoc at Dr. Bruger's lab, and I'll be talking about identifying the molecular mechanisms of HIF2-independent tumor development in KCRS22. So as you probably know, the main driving pathway in KCRS22 is the VHL-HIF2 axis. So upon loss of VHL, HIF2 alpha is stabilized and forms the dimer with HIF1 beta, which drives expression of genes involved in tumor development. However, the advent of small molecule inhibitors of HIF2, which block dimerization of HIF2 alpha, HIF1 beta, it has become clear that not all of the KCRS22 tumors are HIF2 dependent, and there's at least a subset of KCRS22 tumors that are HIF2 independent, and this was published in 2 studies in 2016. So here at the bottom, you can see that upon treatment with HIF2 inhibitor PT2399, even though the dimerization of HIF2 alpha, HIF1 beta is blocked, you can see that the tumor growth is not significantly affected compared to vehicle-treated samples. And this is just for one of the patient-derived xenograft models that we have in the lab, and we have demonstrated that for more samples. And you can see that the gene expression pattern is also not affected. So we hypothesize that there is a VHL-dependent, HIF2-independent tumor development pathway that drives tumor genesis in these subset of KCRS22 tumors. So to explore these factors and pathways, I have developed a few systems to explore what are the unknown targets of VHL. So we have conditional overexpression and conditional knockout systems. And the 2 of them that I'm showing here, they are doxycycline inducible systems. So on top, you see doxycycline inducible system for overexpression of VHL. And we implant these patient-derived xenograft models in mice and look at the tumor growth. And as you can see in the tumor growth curves, upon adding doxycycline to the drinking water, we can stop growth. And that is just kind of proof of concept that this system works. And the bottom is a HIF2 knockout system that we have in a HIF2 dependent line. So we expected that by knocking out HIF2 alpha in a HIF2 dependent PDX line, we would be able to stop growth, and that is what we observe. So these are just proof of concept that our systems are working. So of course, the first VHL target that we wanted to test was HIF1 alpha. So we looked at how loss of HIF1 alpha would affect tumor growth in a HIF2 independent line, XP258. So this is just a tumor growth curve, and as you can see, the HIF1 knockout system or the HIF1 knockout cohort compared to control doesn't show a significant difference in the growth rate. And we looked at the HIF1 alpha at the end of the trial, and then we see that HIF1 alpha is significantly downregulated compared to control. So that indicates that at least in this line, HIF1 alpha may not be playing an important role in tumor growth. And then, as you probably know, there are other non-HIF related targets of VHL that have been identified in KCRS22, including ZHX2 and SFMBT1. And these are more recent studies that came out, and they are known to be, they are reported to be important prognostic factors in KCRS22. So we wanted to test the role of these two known non-HIF related targets of VHL in HIF2 independent tumor development. So this is just so this is just a preliminary data. This is an ongoing experiment. So we use the similar system to knock out ZHX2 and SFMBT1 in HIF2 independent lines. And as you can see in the growth curves, although there is a delay in tumor growth at the beginning, the rate of growth seems to be very similar between control and knockout lines. So we think that these two factors might not be playing an important role here either. And of course, they have to be validated further, and this is just one line. So some of the future experiments that we are working on, we are working with, in collaboration with Dr. Cheng Zhang at UT Southwestern. What we want to do is do a VHL substrate capture experiment. So we would be working on preparing lysates from HIF2 independent tumor grafts, doing a pull down assay in vitro, and doing a mass spec to identify what are the novel VHL binding factors in these tumor grafts. And then of course we would have to validate VHL targets and identify what are the misregulated pathways in these lines. So at the end, I would like to thank my PI, Dr. Brugolis, for his support and advice throughout the project, and Dr. Cheng Zhang for offering a lot of advice on how to set up these VHL substrate capture experiments, and all of the present and past members of Brugolis Lab who have been very helpful. And of course, DOD for providing funding for this project.
Go ahead. Are you okay? Trying to kill people. Quick question. So in terms of when lines become HIF2 independent, are they similarly also TKI independent in terms of from a therapeutic standpoint when you've looked into your xenograft models? And second question I have is, do you think that the development of HIF2 independence is a genomic phenomenon, or is this an epigenetic thing, or is this something that's acquired or something?
Could you repeat the second?
Oh, the second question is, what do you think is the mechanism by which HIF2 independence develops?
Okay. Yeah, well, that's the $1 million question, right? As far as being VEGFR independent, is that your first question? Yeah, so when we look at the rate of growth with sunitinib, I think if you go to one of the slides, we have, can we go back to slide 2? No? Next. All right. So here we have the sunitinib inhibition. So you can see sunitinib and PT2399, they're growing basically identically. So I don't think this is a very so sunitinib is basically inhibiting VEGFR, right? So I don't think there's a significant difference between inhibition of VEGFR in this line and the control line. So if I remember correctly, this is the case for all of the HIF2 independent lines. Now, how they are VEGFR independent, I don't know. And then how they become HIF2 independent, that's the main to be addressed. So yeah, I would say that has to be explored.
Is it still dependent on the VHL being lost, or?
So VHL is lost in all of these lines. So I would say yes. Yes. That was one of the growth curves that I showed, that if you reconstitute VHL, it stops growth even in a HIF2 independent line.
So if I can say something follow-up, when you put VHL back in, you see such a dramatic effect. But when you knock out HIF2, is it correct to say that you're slowing the rate of growth? Because if you look at your last time point on your graph, it seems like the tumors are start growing back. It's heading that way. Would it be fair to say that?
Okay, so you mean the HIF2 knockout in the
HIF2 knockout
dependent lines. So, so one possibility is that because in these lines we cannot do single clone selection, so it's just a population of different. So it's possible that there is a small population with some level of HIF2 that eventually takes over. But yeah, I think that might be the main reason for saying that.
Okay. We don't see anything in the chat. You want to move on to the next?
I mean, our next talk is actually touching on a similar subject. So if there's additional questions and we see them on the chat, we can ask of the two speakers. Okay, I think now it's Shizhuan Liu, who also will be speaking about HIF independent a little bit mechanisms.
Can you present, can you show the next? Okay, good.
Right. My topic is loss of VHL actives. Active, okay. Because the former speaker has already introduced some background, so I will just have a quick introduce. The left panel is the HIF2 alpha sGNRE data, and the right panel is HIF2 alpha inhibitor data. From those data, we can see some CCRCC cell lines are dependent on HIF2 alpha, and some CCRCC cell lines are independent on HIF2 alpha. So those motivated us to look for other targets of CCRCC regulated by VHL. And according to this published data, the hydroxylated HIF will bind with VHL pocket. So suppose other substrates will competitively bind into the same pocket. So based on this, we designed a genome-wide screening strategy to screen VHL unknown substrates. We used 17,000 CDs and we did translation and we got the protein mixture. And next we use VHL to pull down the protein mixture. And in one group, we added Y-type HIF peptides. And in the other group, we added hydroxylated HIF peptides. Suppose the hydroxylated HIF peptides will competitively binding with VHL. So our expectation is that other unknown substrates can bind with VHL. However, in the hydroxylated HIF peptide group, the binding is decreased. So based on this, we identified that the transcription factor SFMBT1 as a novel substrate of VHL. And also the depletion of SFMBT1 can lead to decreased CCRCC growth. And next, we identified the targets of SFMBT1. First, we did ChIP-seq. From the ChIP-seq data, we can see that the genomic distribution of SFMBT1 is highly correlated with H3K4 trimethylation and H3K27 acylation. And these two markers are histone active markers. So that means SFMBT1 can drive genes transcription. So based on this, for ChIP-seq, we focus on the genes that contain SFMBT1, H3K4 trimethylation, and H3K27 acylation binding peaks. And for RNA-seq, we focus on the genes that can be positively regulated by SFMBT1. And for the clinical data, we chose the genes have higher expression level in the tumor tissue. And the more important, these genes expression level should have a positive correlation with the prognosis of CCRCC. So based on this, we got some direct targets of SFMBT1. Here we chose SPHK1 to do some further research. And the deletion of SPHK1 can block both HIF2 dependent and HIF2 independent cell length growth. So in summary, we identified SFMBT1 as a novel substrate of VHL. And in the VHL mutated patient or under hypoxia conditions, the hydroxylation of SFMBT1 will be inhibited. So this stable SFMBT1 will active SPHK1 and then promote CCRCC. And we also tested the SPHK1 inhibitor called PF543. Due to the limitation of time, I will not show the details. And we found that the PF543 treatment can inhibit CCRCC by inhibiting SPHK1. And after our paper, one paper reported that the combination of SPHK1 and HIF2 alpha inhibitors can further inhibit CCRCC. So this is consistent with our data. And we also tried the xenograft model. We found that the PF543 treatment of mice tissues, the treatment of PF543 can inhibit the important factors of NF-kappa B pathway, such as the phosphorylation of IKK alpha, beta, and P65. We also found that SPHK1 can bind with IKK alpha and beta directly. So we will continue to research the underlying mechanism and our other aims for DOD grants. And I'm very appreciative for the help from my mentor, Dr. Qin Zhang and Albert Bowen, aside for the support of DOD. Thank you.
Jim, please go ahead.
Microphone.
Microphone for Dr. Pardoll.
So how specific is the effect of targeting SFMBT1 or the downstream kinase to VHL deficient cells?
Sorry, I cannot hear you clearly.
So targeting SFMBT1 and the downstream kinase is the growth arrest or the reduced proliferation is specific to VHL deficient cells?
Yes, we tested the phenotype and the deletion of SPHK1 can inhibit CCRCC and also the inhibitors. And we also found that the SPHK1 deletion can block the overexpression phenotype of SFMBT1. And we also tested the xenograft model.
Hello. Hey, Suzanne. I think the James' question is regarding whether SPHK1, SFMBT1 targeting will actually affect VHL wild-type cells proliferation or the renal epithelial cell, cell proliferation.
Okay, we tested the VHL wild-type cell lines, KT1 and HKC, and we found that no matter for the deletion of SPHK1 or SPHK1 inhibitor, they will not affect VHL wild-type cell lines. And also the deletion of SFMBT1 have no effect for the VHL wild-type cell lines. Thank you.
I have a bit of a mechanistic follow-up. So if you, I know you did the necessity experiment, you inactivated the kinase and you're seeing a fitness defect in the VHL deficient cells. If you take the SFMBT1 deficient cells and put back the kinase, is that sufficient to rescue the fitness defect over there?
Yes, we actually we also mutated SFMBT1 and overexpress the wild-type SFMBT1 and the mutated SFMBT1. The mutated SFMBT1, we mutated the protein site. So that means SFMBT1 cannot be degraded by VHL. And then we put back SPHK1, and SPHK1 can block the mutated SFMBT1 effects. And we also overexpress SFMBT1 and then depleted SPHK1. And all of these data are consistent with our conclusion.
And sorry, clinically, what is the, have you looked at expression patterns? What is happening to this kinase from a clinical standpoint? What are the clinical associations, if any?
Oh, we didn't take the clinical association. This is just some preclinical data.
Okay.
Another question over here. Dr. Hensky.
I was wondering how many bona fide VHL targets you think there are, how many showed up in your screen, and why did you decide to focus on SFMBT1?
Yes, we got several targets. And also we published one paper about another substrate CHX1. And for SFMBT1, we also tested some other substrates, but we didn't have a good antibody. And for SFMBT1, the antibody is much better, so we chose SFMBT1.
You might have showed this. How much is SFMBT1 upregulated in, for example, in the TCGA of clear cell carcinoma? Is it markedly upregulated relative to other tumors?
Yes, for SFMBT1, SFMBT1 can also have a very significant Kaplan-Meier curve for breast cancer, but we didn't test it, and also for brain tumor, but we didn't test it in that cancer models.
Just a quick question. You know, a lot of targets converge on NF-kappa B, which is, you know, the end of my existence what day, which make me ask, how specific is your finding?
Thank you. So you want me to walk there, Tony? Okay. It's okay. There's no such thing as free lunch.
Tony, is that a question for me? Okay, thank you. Okay, I wasn't expect, but yeah, great. It's a great question. I think that from that perspective, either ZHX2 and at least part of SFMBT1, the actually oncogenic signaling goes through hyperactivating NF-kappa B. So this, we also know that in the VHL now kidney cancer, the NF-kappa B pathways activated in a vitro dependent but HIF independent signaling published by many other labs. So our findings through multiple papers suggesting alternative VHL substrates contributing to this NF-kappa hyperactivation through HIF-2 alpha independent manner. So which suggests the combination of HIF-2 alpha inhibiting strategy with NF-kappa B inhibitors may actually be either synergistic or additive effect in terms of inhibiting tumor growth. This is also one of the directions we're planning to pursue in terms of combination therapy. Yeah, so we do have some NF-kappa inhibitors, one of them being collaborated with our Baldwin's lab at UNC, who is a leading expert studying NF-kappa B. He has one compound, so-called compound A. So we have been testing in different xenograft model as well as mice models. Yeah. Stay tuned. But that's a great question. Thank you.
Can I ask you a quick question? So this VHL E3 ubiquitin ligase machinery is the only machinery involved in the degradation of SFMP3? In the absence of VHL, is there any other E3 ubiquitin ligase can lead to its degradation? Like with HIF, when VHL is not there, other E3 ubiquitin ligases can lead to its degradation. Is a similar mechanism involved here?
For SFMBT1 degradation,
PT1, sorry.
It's the same mechanism with HIF-2 alpha, and we didn't test whether there's other E3 ligase for SFMBT1, so we don't know the answer.
Thank you. Just christen the gene's name. Okay, shall we move on?
Yes, I don't think we can see questions. If there are any questions from the audience. There's okay. So I think we can go on to the next talk. And Pavlos will be leading us talking about hypoxia and tumorigenesis in RMC.
Yeah, thanks for inviting me. When they called me yesterday evening to ask me to talk about this, I said, how long do you want me to talk? 7 minutes, 20 minutes, an hour, a day, a week on RMC? And what do you want me to talk even about? So a pleasure and a privilege, as always, to give some updates on our research on renal medullary carcinoma, RMC. These are my disclosures, none are related to this talk. And as a reminder, renal medullary carcinoma is extremely aggressive, one of the deadliest, if not the deadliest, kidney cancer. And it predominantly afflicts young African Americans, at least in the US, with sickle cell trait or other sickle hemoglobinopathies. It's the sickle hemoglobinopathies that is the connection. We've talked about this, we've done research on this with the help of the DOD, KCRP, to elucidate why it is more common in men, why it is so much more common in the right kidney, and the connection with exercise that we actually described last year in this exact meeting. And of course, RMC, as you guys know, is defined in 100% of cases by loss of the SMARCB1 epigenetic chromatin modifier tumor suppressor. Now, the one thing that is a common theme, every time we come and talk about RMC here, we keep talking about hypoxia. Hypoxia is a hallmark of RMC. Why? Well, at least in part because the sickling of the red blood cells, whether you have trait or disease, sickle cell disease, will increase the renal medullary hypoxia. Even, in our comprehensive molecular profiling we published in 2020 of RMC versus adjacent normal kidney, you see hypoxia pathways definitely upregulated in primary RMC tumors. So here is a big difference between RMC and for sure clear cell RCC and probably some of the other RCCs. RMC arises under extreme hypoxia. Remember, the medulla is one of the most hypoxic tissues in the human body, probably the most hypoxic. So it learns to evolve under actual hypoxia. Clear cell RCC believes it's hypoxic, but not necessarily so. It's pseudo-hypoxia. You lose VHL and then it has blood vessels to help feed it. RMC doesn't need that. RMC evolve under hypoxia. And this is why we believe now RMC is so uniquely resistant to the anti-VEGF TKIs. If clinically you try to give any of the anti-VEGF TKIs, it's the same thing as giving patients water. They just don't work. Zero response rate defined by Nizar Tanir in 2017 and Amishi Shah. And Melinda Shong, along with Gianni Genovese and our group, recently identified that at least one reason why RMC is so resistant to hypoxia is the loss of SMARTB1. In fact, SMARTB1, at least in renal medullary cells, appears to protect from hypoxic stress. And in fact, cells will naturally downregulate SMARTB1 under hypoxic conditions. We have GEM models now of RMC with sickle cell trait. We knock out SMARTB1 and they metastasize thanks to the brilliance of Gianni and Melinda. And we're able to even see that when we implant syngeneic RMC tumors that express SMARTB1 under sickle cell trait, they downregulate SMARTB1 and they turn into big tumors. So there are other orthogonal data showing that. So one of the first targeted therapies to actually work in RMC, we published it in 2021, is erlotinib plus bevacizumab. And this is retrospective data. At the time, when we was thinking about assessment, I thought we were targeting some kind of, glucose metabolism, same thing as the pseudo-hypoxic phenotype of HLRCC. But that's not the case, because if you think about it, we saw responses, they're defined there. What does bevacizumab do? Based on what I told you, it shouldn't do anything, but we didn't have the patients to test, monotherapy erlotinib, versus monotherapy bevacizumab. But we developed a lot of orthogonal data from our GM models and from our PDX models. In fact, Nikki Millward and Manuel Ozambella did this PDX experiment, and Manny was actually disappointed. He didn't know what we were expecting. He didn't know the clinical data. He thought that there should be synergy. There was none. Exactly as we would expect. It was erlotinib alone. In fact, even in 2020, if you look at our GSEA data, we had already seen that RMC appears to upregulate the EGFR pathway. That's what erlotinib was targeting. Now, the funny thing is that once I saw that PDX survival curve, I discussed this with my wife in the dinner cups. And my wife is an inflammatory breast cancer expert. She was one of the people that developed this targeting of panitumumab, a monoclonal antibody against the EGFR, along with carboplatin and Abraxane in triple negative breast cancer. And she showed that they found that inflammatory breast cancer responds remarkably to this triple regimen. And through our discussions, we know there are some commonalities between RMC and inflammatory breast cancer and some differences. But one of the commonalities would make us predict that this regimen could actually work in renal medullary carcinoma. In fact, what she would claim is that when you have a tumor that is EGFR wild type, like RMC, you should not expect TKIs like erlotinib to do better than mAbs. You should expect mAbs to do better. The TKIs work against the EGFR mutants. And so the other thing that was nice about, that brainstorming was that indeed that doublet of carboplatin, nab-paclitaxel, for whatever reason, occasionally can work in platinum-resistant RMC. So it so happened that literally that same week, we had a patient, 68-year-old, on the older side for RMC, metastatic RMC to the lungs, lymph nodes, liver, like legit stage 4 RMC, who had a 6-month response to our preferred first-line regimen, carboplatin plus paclitaxel. Then we put him on our trial, which, by the way, we recently completed 30 patients with RMC, ixazomib plus gemcitabine plus doxorubicin. We already published our data of the backbone gemcitabine plus doxorubicin can work. We will see, we will analyze the signals on whether ixazomib adds anything, but it gave this patient 4 months. But then he rapidly progressed. He progressed to the point that we couldn't give bevacizumab, no way, because he had GI issues. He had a PS of 3. Hospice was discussed, but we said, let's try it. Let's try this triplet on this regimen. There was pushback. It was the first patient that was consented for this. People said, why are you even doing this? But we did it, and there was response. So this guy is now walking and talking and doing great thanks to this triplet regimen, 6+ months response to this. Not saying that this is going to happen with every patient, but this is why now we are investigating further this strategy. So in conclusions, the loss of SMARCB1 protects RMC from anti-angiogenic pathway therapies. We have tons of orthogonal data showing that. EGFR pathway appears to be a therapeutic vulnerability for RMC. And as subsequent steps, we need to understand further, how do the biological adaptations of RMC to hypoxia can be therapeutically targeted? And then, of course, validate the panitumumab triplet strategy to more patients and identify and predict and accommodate for potential resistance mechanisms, phenotypic transformation, heterogeneity, microenvironmental upregulation. That's, by the way, what my wife thinks is the reason why the mAb may be working better, and bypass pathways. So as an acknowledgement, our patients and our patient advocacies, this work happens thanks to the work of patient advocacy and our group in Houston at MD Anderson and throughout the world, and our funding agencies, including 2 DoD KCRP awards dedicated to RMC. And this is how we are able to make this research and help patients. Happy to answer any questions.
Hey, Pavlos, wonderful talk as always. I have 2 questions. One is how hypoxia downregulates SMARCB1. That's number 1. Number 2 is that how do you propose the RMC? There's hyperactivation of EGFR signaling activation.
Yeah, great question. So we know that the downregulation is through ubiquitination. I do not believe that in our paper under revision we mention which one we think is the ligase. We have some candidates, but it's through ubiquitination. It's not transcriptomic. It ubiquitinates SMARCB1, the hypoxia, results of the ubiquitination, and you get downregulation, and that protects from hypoxic stress. With regards to EGFR, so the nice thing about RMC is that it is one of the very few cancers where you have this phenomenon from the same organ, the kidney. You have 2 cancers, RMC and malignant rhabdoid tumors, MRT, that are supposedly defined by the same genetic event, the loss of SMARCB1. So it's a beautiful model to understand, these differential expressions of pathways, etc. So using, for example, MRT as our control, and the other advantage, by the way, is that we have another cancer, collecting that carcinoma, the cousin of RMC that looks like RMC, arises supposedly from the same cell as RMC, but is SMARCB1 positive. So when you compare those 3 cancers, I suspect that the loss of SMARCB1 is necessary, but not sufficient for the EGFR pathway upregulation. And we see that with a lot of other phenotypes of RMC. We're exploring more about this, and so stay tuned, maybe next year.
Pavlos, beautiful work. So stepping away from the cancer, looking at the vasculature that actually feeds medullary renal cell carcinoma, what do we know about the anatomy of these blood vessels? And maybe Payal may have her own view on these things.
Yeah, so one of the things that I learned from Chris Logotheris and Gianni Genovese is exactly to always look at the pathology. It helps immensely. So every patient that I see, I actually look at their slides, it helps immensely connect with biology. So Payal, what do you think?
Yeah, so from these slides, what we see is both these tumors, both collecting duct and RMC, are very hypovascular. So they're just sheets and sheets of rhabdoid cells with hardly any vasculature. So they don't really have, and collecting duct in contrast is more tubular, not vascular, but a lot of desmoplastic reaction, which we don't. And these tumors, RMC, are particularly very inflamed. So yeah, this.
Isn't it beautiful how this pathology connects with the biology? In fact, when they lose SMARCB1, you can see more of the rhabdoid cells, which is a characteristic phenotype of SMARCB1 loss. Hence, malignant rhabdoid tumors. That's another way you can predict. But otherwise, collecting duct and RMC look very similar. You have to have a lot of experience, like you do, to tell the difference. But it's like night and day when it comes to the vasculature compared with clear cell RCC. You have another question?
Pavlos, couple of questions actually. Yeah. So one is coming to the actual biology of the tumor. So in vivo, I think there's a plumbing problem in terms of the drugs not reaching the tumor. And then there's a biological response problem where the TKIs may not just be necessary for function. So if you take your syngeneic lines ex vivo and now try to ask the question whether SMARCB1 presence or absence is going to impact TKI response, do you see any effects?
Yeah, that's a great question. And there are many ways to interrogate this. We haven't necessarily done all of them, but in the experiments that we have done in vitro and in vivo, it appears that the TKIs don't work. They don't help. Now, the question that stems from what you said is, okay, then how does RMC feeds itself? Stay tuned on that.
So just a quick follow-up on the strategic aspect of making the mouse model. If SMARCB1 is going to be downregulated by physiological hypoxia, in the tumor model, even if you put in the wild type or the mutant, both of them should get in the medulla get downregulated. Are you putting in the proteasome resistant mutant over there? How are you doing this?
Yeah. So we have identified a resistant mutation that protects from ubiquitination SMARCB1, and we have done those exact experiments. Absolutely. Yeah. Do you want to?
Without great talk, Pablo. Without claiming too much of molecular knowledge, why not just treat these cells with bortezomib, the proteasome inhibitor, and just see if your hypothesis really stands, where the degradation is truly through the ubiquitination, without necessarily introducing the transfectin with the mutant proteasome?
We did that. It's in the manuscript. So that's how we know that it is through these pathways. We treat them with proteasome inhibitors, etc. Proteasome inhibitors is something that we're super interested in. In fact, our trial is ixazomib, the proteasome inhibitor, along with the backbone of Gem-Adria. So always we try to interrogate the interrelationships between these pathways and SMARCB1 loss. In fact, one of our KCRP awards is this proteotoxic stress that renal medullary carcinoma protects itself from by loss of SMARCB1.
And the follow-up question, now that you are, so you're saying the SMARCB1 levels are somewhat higher during the proteasome inhibition, allowing the cells not to go into this abnormal growth. We still don't know, right, if the SMARCB1 level have any specific connection to EGFR overexpression.
So great question. So one thing I should have clarified. Normal cells, normal renal medullary cells will downregulate SMARCB1 through this process, but RMC itself has lost it genomically. So what happens is that the cells are motivated, and we didn't know that. We had, in clinical cancer research, we published in 2018, our model where there were different steps that would motivate the cells to potentially lose SMARCB1, but now we know that another additional step is this protection from hypoxia. So the cells downregulate SMARCB1, the normal kidney cancer cells, but the normal kidney cells, but they don't lose it genomically. Then the cells that get transformed actually lose MARP1, and they lose it through inactivating translocations and deletions. That's the most common mechanism. One allele is through the loss of 22q, and the other allele is through inactivating translocations.
Thank you. And now that I've shined with molecular biology, tell me how in the world is it 4-1 or 75%, whatever it is, predisposition to a right side? Is sleeping on the right side? You think more people sleep on the right side and they just compress the kidney more with guts that I have? Or
so the answer was given in one of these meetings years ago. This was the first DoD KCRP we got, which was a concept award, was to test that. And the cool thing was, so we postulated in our clinical cancer research commentary that it's the fact that the right renal artery is longer than the left. And what happens is we're thinking, hey, when you have somebody that has lost tons of blood and you want to transfuse them, the blood flow is related to the length of the tube. So you want to have a short tube. That's why we have the cortices, etc., and the diameter. The longer the tube, like an artery, is, the lower the blood flow. This is, in fact, the reason speculatively why cocaine-induced renal artery infarcts happen on the right side 3 times more commonly than the left. Now, the nice thing is that the right renal artery is longer than the left in humans, but it's also much longer in mice. So if that mechanism is correct that we postulated, we should be able to see that. Indeed, we confirmed that in our mouse models, they do have a longer right renal artery than the left one. And indeed, through various orthogonal mechanisms like assays, we have found that the right renal artery, if you put in sickle cell trait, not the wild type, you will see much more hypoxia due to the reduced blood flow in the setting of sickle cell trait. So what that tells us is that RMC is a disease mainly of the microinfarcts from the red blood cell sickling in the sickle cell trait. And what happens is that whether you have sickle cell trait or sickle cell disease, the phenotype in the medulla and the medulla alone is pretty similar. And what happens is that there are 55 times more people in the country with trait. And so if the phenotype is the same, you would expect to see many, 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 many more people with RMC having the trait. That's the connection of RMC with trait. So if you have sickle cell disease, you have similar risk, maybe even more, of developing RMC, but there is this lower number of them.
Pablo, you have 2 questions in the chats, and I promise to show love to the folks in the virtual. So is RMC still considered to originate from distal nephron-based cells, and any role for immunotherapy in RMC?
Great question. So it's a little bit, I would say I'm pretty confident that it originates from the medulla, but the exact cells, I think this is still under investigation. So I will remain a little bit agnostic with regards to the exact cells, but I suspect that whatever those cells are, they're probably either the same or similar to where collecting duct carcinoma also comes from. Any role of immunotherapy in RMC? Unfortunately, I didn't present it here, but Nizar Tanir knows this very well. The traditional immune checkpoint therapies that work against clear cell RCC, they don't work against RMC. Our first clinical trial that was dedicated to RMC tested nivolumab plus ipilimumab against RMC, and we had to shut it down for futility because these regimens do not work against RMC. We have this hypothesis based on data like the ones I showed you, that the potent inhibition of LAG3 may reverse this. So RMC is a very inflamed cancer, but it just has the wrong immune cells. This is the rationale behind our just activated trial which is dedicated to RMC only, where we're using high doses of relatlimab, higher, 3 times higher than what's used in melanoma, along with anti-PD-1 inhibition with nivolumab to see if we can reverse this. And what we do with all of these trials, we get tons of biopsies, blood samples, etc., to prepare ourselves either for success or failure so that we can keep developing this and one day figure out how to harness the immune system effectively against our oops. Okay, mic drop.
And I think with that
Signs. That's a sign from Powell's buff.
Yeah, so I had a question, but I'm going to be a good moderator and end the session here because we're just out of time. So Pavlos, I'll catch you outside. But thank you to all our speakers for presenting today. And with that, we close our session.
Thank you, panelists. Thank you, folks. Real quickly, great quote. I want to say thank you to Dr. Pardoll and Dr. Topalian for their virtual fireside chat. And there's a great quote in there, the power of advocacy is incalculable. I think we're going to be using that going forward. Also, thank you, Hans and Tony, for sort of punting there with that virtual session. And Maria and Nettie, thank you very much for co-moderating as well as the panelists. Quick housekeeping. You do have a microphone on your pod table. It's got a little icon with a voice. If you press that, you can speak. You don't have to wait for a microphone to come over. Folks, virtually, feel free to continue to add questions through the webinar, through the Zoom chat. Lunch is going to be outside until 1 o'clock. It's about 70, almost 70 degrees outside if you want to go down, get some fresh air. But be back here about 1:10 because we're going to start promptly at 1:15 with session 2, which is on novel pathways for therapeutics beyond checkpoint inhibitors. And with that, I know I'm biased here. I'm a big Red Sox fan, but I just want everybody to get out their calendars for next July 13th and 14th. We're going to be in Boston. And that's partially due to ESMO's being held in October next year, as well as the French are having a big meeting, and we didn't want to compete with them. So next year we're going to Boston, the Intercontinental Hotel. You can fly into Logan and take the water shuttle over to the Intercontinental. We're gonna have a big crowd. Hopefully we'll have more people like we did this year. So appreciate it. Take a break, have lunch. Thanks for the morning.